So now I'd like to go over the specific microorganisms that you would see in a soil sample. Okay, microorganisms. I want to start with this slide. You saw it in the last presentation. This is the plant succession above ground and the soil succession below ground. And what I want to point out is, as you see on the far left here, bacteria in that sterile soil. When you first start out, it's like if there's a volcano and you have that sterile soil, the first microorganism that shows up are bacteria. Bacteria needs high light exposure, bare soil, right? You're just bare to the sun. And it starts to be able to function, and plants that can grow in that, pioneer plants is what we call them, the very first plants that show up, can grow in that high bacteria soil. As those plants live and die, and there's a lot of weeds, that grows fast, produces a lot of seeds, right? And can live in bacteria soil. So the plant dies, and now you have some biomass, and that biomass starts to biodegrade, and that material becomes food for the next succession of microorganisms. And so those microorganisms can have a habitat and food and can thrive. And so that's the process that's happening all along under the soil as you move from bare soil to old growth forests. And as we know, if we leave prairies completely alone, they will, as that biology moves, they move into more complex biology, and now more complex plants can grow in that soil, and it will return to a forest situation. So I want to start out so that you can see, and this isn't exactly the fungi to bacteria ratio because it would be very hard to depict that. This is from Dr. Ingham's book, and 10 ways to garden with nature. And so you can see above ground here, the different plants, there's the different successions of grasses that then move to more complicated lower plants, herbs, vegetables. As you go, you get more and more complex to shrubs and small trees, and then larger trees, and then old growth forest. And you see, as you move through here, let me just move myself out of the way so you can see that this, oh, let me go back. This bacteria is getting less and less as you move to the right, and the fungi is getting more and more. And you also see that these other organisms are also still there, even what we would consider plant, disease causing and aerobic. You still want to have all of that food chain present. So we're not killing any of those out, but those are kept in check by the soil biology, the bacteria that are eating those, and they're part of the food chain. So you still see represented those microorganisms. And so that's the full picture. And so now we want to look more closely into the specific microorganisms that we're concerned with in our work in soil biology. And so you see, like, if you look on this slide, right at just before old growth, the deciduous, that is how what Dr. Ingham has said is where Moringa is in this plant progression. And so it can grow, though, in all of these different areas. We just need to get the same soil biology into that as cut and come again. So we talked about before in your permaculture zones, it can be in all those zones, because those zones you would be using that biology for growing Moringa there as well. And as long as you have a dominant fungi, bacteria, fungi to bacteria there, Moringa will thrive. It can also thrive in less fungi. And so in those other zones where it's intercropping, right? And one of the challenges that you're going to see that Moringa will go as a deep root down. And as it goes into that deep root down, it's bringing that biology down there under. So above it can be the other plants growing, the shallower roots, because it's not going to interfere or interact with that fungi to bacteria ratio that is lower than that moringa root. It's going deep down because it's the tap root. So that's what's happening underground. So what you don't want to have at the base is something like grasses that is a high bacteria. So you want to pick things that are actually beneficial in the growing of moringa, like nitrogen fixing things, right? Because nitrogen will be pulled down into those layers, right? As the biology starts to work and get set up and all that crumb, right? The highways and byways. So that nitrogen is pulling down into there and feeding the root. There's a whole system going on. This is what I wanted to make sure that this is a really important visual to have. This is an overview of a close-up for microorganisms. I'm going to start and stop so I can discuss it. So what you're seeing whenever there's the dark aggregate, dark blotchy particles, that's the crumb. That's the soil aggregate that's glued together. And you'll see some microorganisms are shredding that, trying to like open that up, because microorganisms are glued inside there, like they've glued themselves in there. And then you'll see crystals and all different things. And just so you know, this is in a drop of water. We're going to see it later in the microscope section, but there's a millimeter of soil. This is actually what we're going to see as compost. And then there's 5 milliliters of water. That's, you shake it and you get a drop of that. And in that, you're seeing all of that activity, all of that life. So just kind of, think about what you're actually seeing. And also when you're looking down into that scope, you're seeing what's called the depth of field. And that is the dimension of the water that's between the slide cover and the slide. So you'll see sometimes here, they'll zoom out and in and things will come into focus and things will go out of focus. So that's what they're doing. They're looking through the depth of field for just that thin amount of water that's in there. So some of it is going kind of quickly. So I'll start and stop this to discuss it so you can see what's going on. And then these are also repeated. I show them again specifically, individually. I think this is a good overview. So here, look closely for that's a nematode, right, flapping around. This is a rotifer. And as you go through, watch to see that there's like this little vibrating little dots around, kind of sort of moving. Some are stationary. Those little tiny dots are bacteria. Those are the smallest microorganisms. And we aren't able to see them in this magnification. We need to have a much more powerful microscope. This one, I'm going to put it back on as a rotifer. It's actually an indication of anaerobic condition. There's a little pinwheel on one end, and that's how it's moving. So pay attention to, right, this moving around, this flapping around. So these, you'll see them come in and out of the field. This is an amoeba moving through. This is a larva. So a larva, or an insect larva, when you see that, that's also a condition of anaerobic because they're looking for that kind of an environment. If it's too alkaline and too aerobic, that's not a good habitat for them. So, but look again, up in this upper right corner, there's the shadow, and also the bottom corner of microanthropods. So they're thrashing around in there, sharing that food source. That's a nematode. Now, nematodes, you're going to see it specifically, but sometimes they'll glue to aggregates and sometimes they get stuck. And so they're sometimes flapping around that and trying to get loose. And sometimes they're flapping around because they've glued themselves there and they're trying to open that aggregate up so the bacteria will be released from there. So that's what you'll see. And so sometimes they're flapping around so much that you cannot actually identify what type of nematode. So I would take that slide and run a flame quickly under it to dry the water out, and that lack of water in there makes them either stop or slow down a lot, so you can identify it. So sometimes you have to kind of work with what's going on there. And here you see again, the nematode flapping around, going through whipping and trying to open up that soil aggregate. Here, if you look closely, you'll see these tiny little shimmery little dots. That was a microanthropod, which we'll see again later. So just kind of a glimpse of what's to come. Wanted to just show you that. Okay, this is bacteria. Show that, and there's also on this right side, it looks like a testate ciliate, but all the little tiny particles moving around are bacteria, and it's in that soil aggregate, right? All those clumps that are stuck together. And then at the bottom there, you'll see even in this part, there's a section there of fungi. So the scientific way to determine fungi, if it's disease causing or a negative thing to have, is by the width. So and the color. So the color that we want to see for fungi, and you'll see them in further slides, is a tan to brown, and it'll have an outside brown shell, long, it looks like a branch. And then it'll have the septa, these little segments that differentiate that then to a fiber, because they look pretty similar. But the fungi have that septa in there. And then to count and see how wide that is, because if it's really narrow, it could be disease causing. If it's much bigger, it could be disease causing. So 2.5 to 4 point is the dimension. But the dimension is measured by the width of the smallest bacteria that you can find in that sample, in your slide. So you look around and you have to kind of imagine the smallest bacteria, and about how many of those stacked side by side would go across the width of that fungi. That's actually how we are counting and determining, because there's no other measurement when you're looking in there. There are little slides that have little, like a ruler inside there, so you can move it around. But Elaine has said this is too difficult. You waste so much time, and you get to the point where you look at one and now, oh, that's three, you're right, or that's four. That means that you've already seen by many times looking at these samples the smallest bacteria that you've been seeing and measuring that. So pretty soon your eye becomes the scale, but at first you're measuring and counting and imagining, and it takes a little bit of time, but then you get there. You do get there. I'm just going to play that one again so that you can see this lower right corner. There are some parts of some fungi there. But all those little darting around, some look translucent, some are really dark, little ones. And so you would select out that's the smallest one there to use as your measurement. Okay, let's go to the next slide if we can. All right, so the key points here. Bacteria, it's the smallest microorganisms there, or the bacteria. They move around the aggregate, and they're moving in and around, right? And they're looking for food, but they are also somebody's food. And so that movement is kind of a survival thing, where they're darting around to avoid being eaten. Every microorganism lives in a tiny bubble of water. So they're in this situation, they're in a slide that has water. So then we see them floating around in there. But in the soil, they are encased in a little bubble of water, and that's how they move within that. So if there isn't water in those, it starts to dry up, that that outer shell also dries up, can cause them to either die or go dormant. The smallest ones in the field are the ones that we use, right, to measure the width of the fungi, so we can determine if it's a good, a good guy or a bad guy. How bacteria enhance soil biology. So it feeds other members of the soil food web. It helps keep the nutrients in the rooting zone and out of surface and groundwater. So the nutrients are staying right there, right around where the root is. It enhances soil structure, improves flow of water and reduces erosion, compete with disease-causing organisms, filters and degrades pollutants as water flows through the soil.
Over 80% of plants live in a symbiotic relationship with a remarkable organism called mycorrhizal fungi. Through this symbiosis, the fungi and the plant naturally develop a lifelong bond of mutual benefit that's existed for over 450 million years. Mycorrhiza is the term used to describe the symbiotic relationship between mycorrhizal fungi and a plant's root system. First, the spore of mycorrhizal fungi germinate in the soil and make their way to the nearest roots. The roots are then colonized by the fungi, and mycorrhiza are established. The fungi penetrate the root and create an internal network of fungal structures inside the root cells, where the plant and mycorrhizae exchange sugars and nutrients. Finally, the hyphae continue to develop outside the roots, forming an extended network of fine filaments, which cover up to 700 times more soil area than the plant's own roots. This secondary root system draws in extra beneficial nutrients and water, supporting a plant or tree for its entire lifetime.
So this it's such a good example of it, an explanation. I couldn't have done a better job. So we see that the spores are there in the soil and they emerge, some go inside the root, some are living on the surface of the root. The ones that go in create this symbiotic relationship that's ancient, as you heard, and then the ones on the outside start to create the mycelium, right, that stretch out and create that massive root system additionally to what the plant has. So if every plant could only absorb the nutrients just at the base of wherever their roots are, they would have a very, very hard time surviving. There's no way to go once they've eaten all of that, that bacteria is there and it's gotten fully fed and there's nothing more. How would they continue to eat, right? So it's that constant movement of the fungi moving out, bringing in more bacteria coming in, and that whole cycle happening that stays right at the root, where the root needs it, and the root then decides when it's starting to send out exudates and then pull in nutrients and then send those sugars and carbs out, those cookies and cakes, which is what Dr. Ingham calls them. And then so that symbiotic relationship and that biological messaging is what's happening at the root of every tree and of every plant that we grow. So when you understand that, then you can see the importance of fungi for all root systems that of any plant you're growing, that it's going to be far more nutritious because it's going to have the reach of as far as that fungi can go. And so every time modern farming tills that up, it's breaking that entire habitat and it's taking up those root spaces in the soil that were created by the previous plant, whatever that was. And everything starts over. We return to zero. So in that plant succession and that soil succession, every time you till, you go back to zero. And so all that habitat, that food cycle has to regenerate again. And it does. And as long as we're not harming it, doing this, that 80% of plants have a symbiotic relationship with mycorrhizal fungi. So you can imagine that 80% of all plants, right? Symbiotic relationship has existed for more than 450 million years. Mycorrhiza is the term that describes this relationship. So there's mycorrhizal fungi, that is the physical one, and mycorrhiza is the relationship. Spores germinate and more into the roots, move into the roots. They colonize inside the root, exchanging nutrients, and fungi outside the roots create a root system covering 700 times more soil area than the plant could produce by itself, could reach by itself. And this is how fungi enhance soil biology. Decompose complex carbon compounds, improve accumulation of organic matter, retains nutrients and fungal biomass, reducing leaching of nutrients out of the root zone, physically bind particles into aggregate, are an important food source for other organisms in the soil food web, improve plant growth when mycorrhizal fungi become associated with the roots of some plants, compete with plant pathogens, decompose certain types of pollutants. A lot of benefits besides just being that root zone. Here are some pictures. That I was describing before, these septa. In this bottom right corner, you can see a really good example. There's the fungi, the hypha, the strand, and it's the outer edge is brown. And if we could see bacteria in here, we would start to be able to count how many of those would it take to go across the width of that. And that septa is what lets us know this is not a fiber, some type of fiber in your sample. And then up on the top, you can see there's the aggregate and that fungi is working through there. And on the other two, that's how you can see the reach of the fungi, the mycorrhiza, and the mycelium that's moving out from the root zone out into the area on all sides, in all directions. So it gives that benefit. Beneficial fungi are characterized by tan to brown sides with segments called septa. Fungi attached to the root systems, giving the plant a bigger area to draw nutrients from. Beneficial fungi are tan to brown in color, are 2.5 to 4.0 bacteria width of the smallest bacteria near them. So that's the piece that you really want to understand. How that's happening. If you go into the microscope world, which I do recommend, but it's its own journey. And this is a little video on that. Look at this. So you can see sometimes the microscope goes in and out of the depth of fields where different things come into focus and other things become unfocused. That's the depth of field. So you see there's some movement, and down below there's bacteria moving. If you look closely, all of that is actually moving. If you really pay attention, it's all in motion. It's all swimming in this little bubble. So all of that is the food source. The fungi is eating as well. And it's creating this habitat there. And you saw the aggregate and also these like crystal looking parts. Those are the sand, silt, and clay particles that you'll see. And so this was just really isolated fungi. I thought it was a really good example. Yeah, you can look at this again and again if you want. It's just amazing. The next one, oops, go back. So we're going to look at protozoas. They're it's a major group based on how they move, their shapes and how they move. So this is flagellates, ciliates, and amoebas. Protozoa play an important role in mineralizing nutrients, regulating bacteria and algae populations. They maintain an ecological balance in the soil when they graze on bacteria. Protozoa stimulate growth of the bacterial population and decomposition rates and soil aggregation. An important food source for other soil organisms and help to suppress disease by competing with or feeding on pathogens. They influence the bacterial composition and bacterial population in the rhizosphere. Here are some samples.
Which is the general name for single-celled eukaryotes, are actually loosely classified by their style of movement, with cilia, flagella, and pseudopodia. We're going to start with the shapeshifters of the microcosmos, amoeba. These cells can change their shape as they wish, allowing them to extend parts of themselves into features called pseudopodia. These extend in the direction they want to move and then solidify as the cell moves into the newly occupied space. Pseudopodia is Latin for basically false feet, and these extensions, which are half reaching arm, half cell body, are how these crawlers move and hunt. The secret here is that the cytoplasm of an amoeba can be easily changed from a fluid state into a solid state and back again. When an amoeba moves, liquid cytoplasm flows through the center up to the tip of the pseudopod and then gushes to the sides, where it becomes more of a solid gel, allowing the cell to lock into its new location. Predatory amoeba create these extensions in all directions to trap prey between them. Once the prey organism is surrounded by the pseudopodia, the amoeba simply swallows it. Though they look like rods sticking through the surface, they're actually extensions of the cell membrane wrapped around rigid tubes called microtubules that are anchored in place. However, hydrodynamically, they're very different. There are usually an easily countable number of flagella per cell, whereas ciliated organisms have huge numbers of cilia. The word cilia actually comes from the Latin word for eyelash, which makes sense as you can sometimes see them ringing a cell as our eyelashes ring our eyes. But in those cases, the cilia actually often cover the entire cell. They're just harder to spot against the background of all the stuff on the inside. Cilia move organisms by beating in waves over their surface. A cilium, which is the singular of cilia because Latin, has only two possible positions. During the effective stroke, the cilium sticks out perpendicular to the cell, and during recovery, it folds back towards the cell's surface. In most ciliates, the cilia are arranged in rows, and the cilia of a row don't move all together at once. When some of the cilia are in the effective stroke, others are in the recovery stroke. And in some ciliates, you can see the wave pattern this creates. And these waves are basically grabbing on to the sticky, viscous water, yanking the cell through. One added complication here, some ciliates have dense bundles of cilia called cirri. Cirri are used to walk on a solid substrate rather than pulling the organism through the water. Though it's the same structure, it is a bit of a different system of movement. Now, on to eukaryotic flagella. The motion of most flagella is characterized by this long wave-like beating pattern beginning from the base of the flagella and moving out to the tip. We said nothing in this entire video about the marvelous movements of prokaryotes like bacteria, and they have systems every bit, if not more ingenious than their more complex eukaryotic friends. But they are also tiny. So tiny that these structures can't be observed by microscopes like the ones we use.
Here is, I had to show this because it's, we saw it before, but this one was just as it's eating. It goes round and then, as I was describing before, it opens the mouth, there's all the bacteria that's right there, and then it draws it in and eats it. And I don't know, these are like, I don't know how big we can say these are, but they're just the tiniest little living things. And this is, like I said, this is the smallest amount of a sample to create that. And it ate, and it goes round again, and then as it opens up, see what the bacteria that's in there came out. And so that's kind of the life of a ciliate. And this is how protozoa enhance soil biology. Release nutrients in microbial biomass for the plant use, increases decomposition rates and soil aggregation by stimulating bacterial activity, prevents some pathogens from establishing on plants, provide prey for larger soil organisms such as nematodes. And here are nematodes. So let's take a look. Those are those flapping around little worm-looking things. Elongated cylindrical worm parasites. So these also exist in all different forms and sizes. Some are parasitic. But this is it just flapping around. It could be stuck or it could have glued itself. And if you look in there, all the little tiny, gently vibrating things, there's the mouth and the foregut, and it's slowed down enough that you can probably identify the mouth part. This is at 400 magnification. You see, it looks like it's just using that to whip itself. And this is where I would say, like, let's slow that down, and we could dry that slide out a little bit, and it would slow it down. How nematodes enhance soil biology. They regulate the population of other soil organisms, mineralize nutrients into plant available forms, provide a food source for other soil organisms that influence soil structure, consume disease-causing organisms. And this is what I described. These are the mouth parts. So this is how they would categorize the different types of, it's a bacterial feeder, fungal feeder, a root feeder, predatory, and omnivore. So if it was slowed down on those slides, we could have identified it by that mouth part. And then we would know what activity it's doing, who's its prey, who's its food in that sample. Microarthropods are now getting to the larger scale, and they're the threshers and movers, and they pull stuff apart. And this is just a really good example of it. You see the soil aggregate there, and it's moving around, and decides when it's going to eat, and just starts to like chow down on things. And so you can see that these little microorganisms are similar to things that we see above ground, right, in the next trophic level. But these are the microscopic versions of those same animals. So this looks like a beetle or a mite that we would be able to see with our naked eye, right? Here's another one that's more active. It's thrashing around, and it'll break up that soil aggregate and eat that, the bacteria that have lodged themselves inside there. And it also is making has a waste product, which is bigger than the others, also full of nutrients for other bacteria as their food source. And so that whole part of the food chain is going on. As you can see that we can identify these kinds of creatures above ground, like nematodes look like earthworms. Earthworms are the bigger version would be snakes, right? So we see all that repetition in nature. And so you see these cylindrical looking things that could be fungi, but I don't see any septa. And so those some other, the one that's close to him, that looks like it has septa there, because they could be little crystalline shapes of things that exist there. So you can look around, and all around him, this little guy, are bacteria that are swimming around just like a sea of food. And it also, the microarthropods eat the fungi. And so you can see it could have been breaking that fungi up, that you see pieces around, and it's moving in that bubble of water, right? So all that's aggregate there is not, now remember how small a sample this is. So in that bigger soil sample, or the compost, there's, maybe thousands of these. And what we would be doing is looking at and counting how many we see in that drop. And if we diluted it 1 to 5, then we would multiply that out to, and to more bigger and bigger to extrapolate what amount you might have in a 3 by 3 cubic compost. And so you're taking a small sample, but then you're saying, okay, this is representing the whole population. So if you see 2 of these and 5 of those and 10 of those, then you're multiplying it out to say how many of those they are in that huge, above ground pile that you have. So that's the point. And then in Dr. Ingham's work, she actually has the spreadsheet she developed so that you start to list those. You just count 1 and 2, and as you see them. And then at the end, it tells you that what your population is when you put in your 3 by 3 by 3. And then so the math is already done for you. So it's really great service, because you don't have to then figure it out mathematically. But you can see that this is just all the life going on in that soil beneath your feet all the time, day and night. Nature working for us. If we employ them to be doing that, it's the nutrient cycling. How arthropods enhance soil biology: improve soil structure through burrowing and the creation of fecal pellets, control disease-causing organisms, stimulate microbial activity, enhance decomposition through shredding of large plant litter and mixing the soil, regulate healthy soil food web populations. So it's having a different, more complex diet of the things that it's eating. And so the waste product is going to be more complex. And so as you go up the chain, those bigger organisms that are eating all those small organisms still are transforming those organisms into nutrients. And then if somebody eats the arthropod, then that crunch, the carbon that is the shell is released, all the fluids inside of the water that's inside the body, and all the nutrients inside that body are now released, and that's a part of that nutrient soup that's going on. So that's the food for all the other nutrients. So you have all that happening. This I want you to watch on your own. There's also a sheet that is there in your advanced references. This is a simple way to take us to do a soil texture test, so that you can see the percentage of soil to loam to clay. It's a really good way. We do this during the course. We get a sample, we shake it up, we let it settle, and then we measure these amounts, and then we put it on that chart so we can identify what texture of soil that you have. This one, I'm not going to play it. You can watch that on your own. What this is, these are penetrometers that he's demonstrating there. And the penetrometer is something that we use to determine how deep your soil compaction is, how far from the surface of the soil. So it's a tool that you press in. I mean, there's different ways. There's simple ones, just like a screwdriver. It's not going to tell you the pounds per inch measurement, but if you have any long, I mean, if it's a short screwdriver and you go down 2 inches and you have compaction, there's a lot of work to do. So what you want to see right when you begin is to have that penetrometer tell you how much soil you have above a compaction level. And so you press that down, and the gauge tells you pounds per square inch, how much pressure that it goes down, that it needs to go down until it stops. And then you have a measurement on there to show, okay, that's 12 inches or that's 3 inches. And then you know that amount of soil that you have. And over time, as you employ these soil biology practices, you're going to go to that same place, and you're going to put that penetrometer down, and you're going to measure how you're doing in your progress there. So it's a really good and simple tool. And you don't have to know pounds per square inch. That's not necessary to know. It's just that it gives you more information. But if you just take any sharp tool and you go down and you measure that, and then over time you measure it again. So it's just a simple way. But watch that whole presentation. I had to include this when I found it. It's just it's a way to measure soil biology without really any instruments at all. You're not getting a clear measurement of how many fungi to bacteria and all of that, but it was so clever. And I just thought, yeah, when you don't have any tools, use this. And it's a cute, clever saying, you know, when you soil your shorts, that means you've pooped in your pants. That's an American, I don't know if it's English even, but it is American. And so other cultures, you might not know what that means, but it's a kind of joke. When to soil your shorts, sometimes you get so scared, you know, soil your shorts. So what this is, a simple way, they take these underwear, and he goes, I think he puts out 60 different, this is a big area, and he has 60 pairs of men's underwear. So you want 100% cotton. So you can do this, of course, with any cotton. And you bury it, and with some little flag piece above ground so you can find it again, or a flag or a rock. And then you come back in a month and you look and see how much of that is left, how much has biodegraded by the soil biology that's in the soil. And so sometimes, you know, he shows that I pull out almost the whole pair of underwear. That means that we could just wash it and wear it again, or wear it. That means you do not have the soil biology needed to biodegrade that, because that is the food for microorganisms, specifically fungi. So watch this. It's a clever little thing. I just had to include it. Todd Harrington's, he's one of Elaine's students, and he has two projects that I've included. He has many. He was in the soil food web summit, soil summit. Oh, I can't get it out. And he just goes over really clearly what we need to do with soil biology, and then he shows. It's in your advanced references, or more soil biology references, that one of his projects that he did on Governor Island. And it's phenomenal when you see this at work. And you can search on YouTube for more of his videos. He's currently a consultant and has a lab in Boston, I believe, and just a wealth of information, and just provided so much guidance for people during the Soil Summit. And so that you'll see that. And there are a lot of examples of the success of this work. And as we go through, I'll have more resources, and you'll be able to see. It's just a point that I have to say, okay, where do we stop? Right? So these are case studies. This is the Governor's Island of that's his project. And this is Paul Stamets. This is a TED Talk. Paul Stamets he's my hero. He is the fungi king of the world. He has brought us so much information about mushrooms and mycelium. And I still, even last week, ordered from his website to get mycorrhiza, the mycelium for putting out under my trees and my plants. And there's 3 different types on his website, fungi.com. And it's one is for trees, one was endo, ecto, you know, so you'll learn all about those terms. One is for when seeds germinate, one is for when you have vegetables, one is for trees. And so you'll learn a lot on his site. But this is an amazing thing that he's put together, 6 ways to save the planet, and he shows and demonstrates with the work he's doing. Now, I have followed him for 20 years. And in that period of time, he has gone from just having this small little business to this so much research and proven things that he can show we can heal the planet with fungi, with mushroom. The whole mushroom world is here to save us. We just have to tap in, and he has. And his life story is quite amazing too. If you just research him and find out about him, he does also Bioneers talks and podcasts, and he's just sharing so much. He wrote a book, Fantastic Fungi, and it's actually a movie that he actually co-wrote it, I believe, with Louis Schwarzenberg. And that's a video that you should watch. It's fantasticfungi.com, I believe is his site. And together they really showed the world of mushrooms and how powerful they are in our lives. So I put it in this section so that I could emphasize it and also encourage you to watch it. These next three are the microscope setup that Dr. Ingham does. This is Dr. Ingham. I believe this is even at her house in Oregon. And so she's showing you, I have this, not exactly the same, but very similar microscope and that we use for looking at these organisms. So she gives such a good, and of course she's the expert, so she gives these next three. So watch these if you are interested to get into the microscope part of this. It's fascinating. This is the fascinating part of my work. I just love the ability to see things that you never ever thought possible, and you don't even think about this world. And there are a lot of resources I've put on more soil biology as a file, and also there'll be more in the advanced references. There are so many, you kind of have to take your time going through, but I just wanted to put as many as possible. And in there, look for the ones that are Suzanne Simard, How Trees Talk to Each Other. She is proving how all the fungi in a forest communicate, and the mother plant who can identify that her own babies around her and feed specifically those trees. So it's a fascinating world. It just gets bigger and bigger the more people are involved and are sharing their research with us. I just feel so fortunate to have come upon this. It's a big part of our work. I feel because in the growing of Moringa with the soil biology, we are participating in the healing of the planet. And as you can see, as we went through, the cleaning the air, cleaning the soil, cleaning the water, the vital things that we have on this planet, and the way we grow Moringa is actually engaging that whole system. So what growing Moringa is, we thought, oh, I'm just going to plant a seed and grow a tree and harvest it and eat it. No, we're actually embarking on dealing with so much of the issues that we have in our environment and all of the contamination. And we know we have what's needed and the information, and the more you learn about, you will find your part in what that can be worldwide, in the worldwide movement of Moringa. And the growing of Moringa, the best way is to engage all that soil biology.